the peptide bond. Okay? So of course we have an isolated amino acid. We have amino acid 1 and then we have amino acid 2, but we care about peptides and proteins, so the actual chain they make. Okay, so the peptide bond itself, it's formed through a condensation reaction. So this is meaning that when these two come together, we're going to pop off water. Okay? This is sort of a different situation from hydrolysis. We need a water to come in and break whatever we're doing in, say, ATP. Okay. So this reaction is happening between the carboxyl end of one of the amino acids and the amine end of the other amino acid. And it's going to result in this peptide or amide bond here between the carboxyl carbon and the nitrogen, okay, our amine nitrogen. This is where the peptide bond is. Okay, so this was alpha carbon from amino acid 1, peptide bond, alpha carbon for the next one. Amino acids linked into a peptide, or linked by peptide bonds, are called residues. Why do we call them residues instead of amino acids anymore? Sure. That's exactly right. They no longer have their charges anymore. Okay? The ends are going to still be charged, but when you have a long string of these, and I say I have a 50 residue peptide or 100 residue protein, all of the things in the middle no longer have acid functionality anymore because they no longer have charge on them. Okay? So we'll start calling them residues. So again, our peptide bond formation also results in the formation of an H bond donor. Our amide nitrogen here now has a hydrogen on it that can participate in an H bond. And our carbonyl, this is now a carbonyl carbon. Again, the names are not so important to me unless you like to memorize them that way. And this one can now, this oxygen here can now serve as an acceptor to participate in a hydrogen bond. And then this green sort of demarcation here is named the amide plane. And the way I like to think about it, it also, this is not a good drawing and I will show hopefully a better figure later. I also link to some 3D, like you can manipulate them in space. Please click on those and try them because like the 2D versions are just not super helpful. And my verbal explanation of what's in a plane, also not great. When you can kind of spin the model yourself, it's nice. These alpha carbons and these adjacent amino acids are also in the plane. Okay, there are 6 atoms that are all in the plane. Picture it flat with the board, and then these R groups could go in or out of the board. Okay, on either side of the 6 atoms that are stuck flat together. Okay? And there's more figures where this comes up, and we'll talk about it a little bit more. So in the polymer chain, what was the backbone sequence for a nucleotide, for our nucleic acids? How does the backbone go? Sugar, phosphate, sugar, phosphate, sugar, phosphate. Okay? So the repetitive sequence for the backbone of a polymer chain is nitrogen, alpha carbon, carbon, nitrogen, alpha carbon, carbon, nitrogen, alpha carbon, carbon, right? That's our backbone. And of course, hanging off the side is our side groups. So DNA, we sort of write it, read it, treat it. We have a particular directionality, okay? 5' to 3'. Proteins also have sort of a convention for this. We're going to always orient things from the N terminal, our amino or amine terminus, N side, to our free carboxyl group, our C terminal, C terminus, D side. So we're going to write things from N to C. It's always the orientation that you'll see them in. So if you were just given a list, again, of just sort of names of amino acids, you would still understand that the amine group's assumed to be on the left side. And the sequence of amino acids is called our primary structure. And our primary structure is very important to dictating what types of secondary structure, what type of folding we'll get. In the double helix, did it matter what order of bases we have for making a double helix? No. The double helical structure is highly conserved, repeatable over and over every time. It's always going to form a double helix unless some sort of base has been swapped out and we have some mispairing going on. Then we might have a kink in our structure. Then it might not be favorable enough to make it But if things are lined up the way they should be, it doesn't matter if it's AAA, GGG, AGU, UT, it doesn't matter. That was RNA, because I did use, whatever, you get it, okay? So again, the 3D structure of DNA does not depend on primary sequence. For proteins, it's very important to what it can do sort of in terms of secondary and tertiary structure. And just really quickly, why do you think this particular little sequence here has the phenylalanine and the methionine drawn like this? Sterics, perfect, okay? Everything is not all on one side, because that would be, there's gonna be some steric clashing going on. This is a summary slide. I don't know that there's anything new on this slide. That seems like a reasonable time to take a break, right? I'll leave this hanging out. So let's take like 5 minutes, and then we'll keep going.